The tertiary structure is determined by how the secondary structures interact with one another. And when we're looking at tertiary structures, it's really important to understand the difference between nonpolar side chains, which are hydrophobic, and polar side chains, which are hydrophilic. These proteins are going to be in many of them in aqueous environments in the cell or in the blood, and the nonpolar side chains are going to be repelled by these aqueous environments and turn into the interior of the protein and interact with each other rather than the aqueous environment. Polar side chains, on the other hand, are going to be attracted to aqueous surroundings since water is polar, and they will appear on the surface of the protein. So tertiary structures are stabilized by many different types of attractive forces between the side chains themselves and also between the side chains and the environment, especially, if we're talking about in a cell that's going to be an aqueous environment. So let's look at this graphic here, and it shows some of the stabilizing forces that occur in the tertiary structure. We have hydrogen bonding, which occurs a lot between the polar side chains. We also have something known as the salt bridge, which is the interaction between oppositely charged side chains. So basic polar side chains have a positive charge, and acidic polar side chains have a negative charge. So it's a salt bridge there. On the interior, we have these hydrophobic interactions which occur between the nonpolar side chains. And we also have this disulfide bond here. So tertiary structure interactions are many. We have the nonpolar interactions that occur between the hydrophobic side chains. And as I said earlier, they tend to aggregate in the inside of a protein because they are repelled by the aqueous environment. Polar interactions, which occur through a variety of interactions known as dipole-dipole, ion dipole, and hydrogen bonding. So we see hydrogen bonding occurs quite a bit. Then salt bridges, which occurs anytime we have ions in the side chain, and then disulfide bonds. So that's going to occur anytime we have two thiol groups interact, and so thiols are found in the amino acid cysteine, and they're going to get oxidized, and when they oxidize, they lose hydrogen and form this disulfide bond. So those disulfide bonds are definitely stabilizing forces for the tertiary structure. There are two main categories of tertiary structure. We have the globular protein, which fold into sort of like globes, if you will. So a compact spherical shape with the polar amino acids on the surface and the nonpolar amino side chains in a nonpolar core. Enzymes and many proteins that are found in the cell are globular proteins. On the other hand, another type of tertiary structure is the fibrous proteins, and these have long thread-like structures made of aligned helices, which form very durable structures. So, and fibrous proteins tend to be insoluble in water. So, two examples of fibrous proteins include alpha keratin, which is found in wool. It's also found in the beaks of birds as well as in our fingernails and our hair. And then collagen, which is found in connective tissue and skin. And gelatin, actually, or jello, is derived from the collagen found in the skin of animals.